Okay then. Any doubt in the last session, guys? Anything?
No, ma'am.
Did you install the tools?
Yes, ma'am.
Prajakta, are you there? Hello.
Hello, ma'am. Ma'am, actually, yesterday I haven't joined the lecture because I didn't receive the link for the meeting.
Okay.
So I joined today only.
Okay. Do you have the WSL, ma'am, in your system?
What's ma'am?
WSL, the Linux?
No, ma'am, no.
Okay. So you enrolled right now in the DNSSEC?
Yes, ma'am, yes.
Okay. So, ma'am, please ping me tomorrow before the session because we have to install the WSL also in your system. Okay.
Okay, ma'am.
Yeah.
Okay.
Okay then. Yeah. Can you see my screen?
Yes, yes.
Yes, ma'am.
Okay. So there are some important terms in NGS. So first is type of sequencing. Can you tell me what type of sequencing are there?
Like the first, second, and third generation sequencing.
Mm, no. So there are basically two type of sequencing, or you can say the raw data which we are getting in the form of single end data and the paired end data. Okay, or you can say pair end sequence. So what is this single end sequence? Have you ever heard about this before?
Ma'am, is it based on the a type of adapter that is attached to the reads?
Yeah. So let's see what is the single end first. So in the DNA there is two strand, forward and the reverse, right? So for example, this is my forward strand and this is my reverse. So in the single end, what happened? At one side, the adapter is ligating and the sequencing machine, it start reading from this direction. And after reading this entire forward strand, you will get what? The forward file. But if the adapters are ligated towards this reverse strand, it is going to start reading from this direction, right? And here you are going to get your reverse file. So in the single end, what happened? The adapters are ligating at one end and sequencing it is start reading from this particular direction, and it is going to generate either the forward file or the reverse file. Whereas in the paired end, what happened? The adapters are ligated towards both the ends and it will start reading from this direction also as well as this direction, right? So here in this paired end sequencing, you are going to get two file here, that is forward as well as reverse. So this forward file will be denoted with the id underscore 1 and reverse file it will be id underscore 2. So wherever you will see underscore 1, it is going to tell you this is your forward file and underscore 2 represent your reverse file. So that means in the paired end, what happened? The adapters are ligated towards both the ends, and you are getting two files at one time. Whereas in the single end, you are getting only one file at a time. Got it? The difference between this single end and the paired end?
Yes, ma'am.
Okay. So now next term is the depth. Have you ever heard about this depth word? Can you guess this depth word? What is that? Any guess?
Number of reads sequenced at a given time.
Okay. Prajakta ma'am? So depth means how many times the particular base is called, read, or sequenced by the machine. That is the depth. Okay. So for example, this is my reference genome, and I'm going to align my raw data on this reference genome. Okay. So these are nothing, these are just the reads that are aligning on your reference genome. On this reference genome, already you know what is the base is present here, right? So for example, on this position, you are having a adenine here. And at this position, what we saw, there is a adenine. So can you tell me how many times this adenine is called by the machine here? Any guess? So you can see on this particular position, there is a adenine and machine is sequencing this particular base here. Okay. Adenine 1, then 2, 3, 4, 5, 6, 7, 8, and 9, and 10. So 10 time the sequencing is reading what? The adenine here, right? So for this particular base, your depth is what? 10x. Are you getting this? Next, for example, now you have to tell me there is a thymine here, and now for this particular position on the reference genome there was thymine, right? By aligning your raw data, you can see there is a mutation also. The T is getting replaced with the G here. And can you tell me what is the depth for the guanine here? Tell me
One.
how many times the
One.
guanine is read.
One.
Huh?
One.
Why one, ma'am?
It is present at the place one. No.
No, 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 no. See, I will just tell you first for this, and then you have to guess for this second one. Okay.
Okay.
So here, this is on this position, ma'am. There is a adenine, right? So I'm telling you, all these 10 reads here at this particular position, they are calling the adenine here. So here, the adenine is called 10 times here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? So all of the base that is present here, that is the adenine here, right? So that is why for this particular position, the depth is 10x, ma'am. Now I'm telling you, on this particular position, on the reference genome, there is a thymine, and you can see out of 10 reads, only 4 reads. 1, 2, and 3, and 4. 4 times the guanine is called by the machine here, right? So that means for this particular guanine, the depth is your 4x. Are you getting this now,
Mm.
Moira?
Yes, ma'am. Yes, ma'am.
Okay. Now again, now you can guess me again. Okay. I will just put an example. Again, there is a adenine at this position, and here it is guanine. Now, Moira, can you tell me what is the depth for the guanine here?
7x, ma'am.
7, 1, 2, 3, 4, 5, and 6, Moira.
Oh, sorry, ma'am. I counted the previous one also. I'm sorry, that was my
Okay, okay.
mistake.
It's
I
for
understand.
reference. Yeah, 7, right? Now, Prajakta ma'am, can you tell me what is for
Ma'am.
this adenine here?
2.
Right, 2x. Okay. So did you got this depth term?
Yes, ma'am.
Okay. Next is the coverage. So coverage means how many reads are able to cover your reference genome. Okay. Again, this is my reference genome here. And reads are aligned on this reference genome. So you can see for this particular example, the 50% of the coverage is there, right? So because the half of this, you can see the reads are not covering the reference genome here, right? 50% of the data it is missing here, correct? Right? Coverage means how many reads are able to cover your reference genome. So coverage should be 100%, right? Because we want to cover the entire reference genome. Here in this example, you can see only 50% of the data it is covering, right?
Yes, ma'am.
So we don't want to miss our data. What we are going to do, we will increase the depth. So by increasing the depth, the number of reads will get increased, and it is going to cover your reference genome. So that means by increasing the depth, coverage is also getting increased. Or you can say depth is directly proportional to coverage. Are you getting this?
Yes, ma'am.
Moira,
What did you ask, ma'am?
did you got this coverage term?
Yes, ma'am. Yes, ma'am.
Okay. So next is very important, that is the Fred quality score. So Fred's quality score means it is assigned to each and every base which is sequenced by the machine and every base by the machine has some quality score which is going to tell you accuracy of the base which is called by the machine. And the standard cutoff for this is greater than equal to 30. So in this Fred quality score, what happened? Each and every base it is sequenced by the machine, and the quality score it is given to each and every base, and the standard cutoff should be greater than equal to 30. So let's discuss why 30 here. Can you see this table?
Yes, ma'am.
Okay. Now you can see this is your Fred quality score. If your Fred quality score is 10, that means 10 time the machine is calling the base, and out of 10 time, 1 time the base was incorrect, and that time base call accuracy is 90% here. Did you got this? For example, 10 is your Fred quality score, and here what happened, 9 time it was reading a adenine here, okay, 9 time, and 1 time it detecting the guanine, right? So there is an incorrect base, right? Are you getting this point?
Yes, ma'am.
Yes, ma'am. Yes.
Okay. For example, again, this is my reference genome. There is a 10 reads. So 10 time it was reading, okay? And what happened? The 9 reads were showing the guanine, sorry, guanine here. Okay, on this reference genome there is also guanine, and one time here there is this what, the thymine. So this is what the Fred quality score here. Okay, you can see one time the base was incorrect here, it is causing the T here, right? If you increase to 20 here the Fred quality score, then 100 time the base is read by the machine, and out of 101 time the base was incorrect. That time the base call accuracy is your 99%. Then if you increase to 30, the 1000 time it is reading, and one time the base was incorrect and it is giving you 99.9. So why we have taken the standard cutoff as the 30? As you can see from this table, by increasing the Fred quality score, it can be 40 or the 50. You can see the base call accuracy is still the 99.9. Can you see that? No Fred quality score, it is giving you 100% efficiency here, right? So that is why we are taking the standard cutoff as the greater than equal to 30 here for the Fred quality score. Did you got this point?
Yes, ma'am.
Any doubt?
No, ma'am.
Okay. So there are some files you might get in the form of BAM and SAM file format. So in this format, this file, it is containing the information containing the complete mapping information. So this BAM file stand for binary alignment map, which is your computer readable file. So you might have heard about the 01, right? 0101, right? That is the binary language that we cannot read, but the computer, it is going to read that. Okay, the BAM file. But if you want to read the file, what is the content that is contained here in the BAM file, you need to convert this BAM into the SAM. So this SAM file, it is what, the sequence alignment map, which is the human readable file. Okay, so the ATGC format, you can read it, right, with the help of SAM file. But if you want to run the tools further, that time you have to provide the BAM file. So this is the computer readable file. So did you got this, the difference between the BAM and the SAM?
Yes, ma'am.
Then in this DNSSEC, you are going to get this BCF and VCF file. So again, this BCF file is your binary calling format, and whereas this VCF is your variant calling format. So this file is very important in the DNSSEC, because in this variant calling format, you are going to get the metadata line, In the metadata you will get all the instruction of your tools, whatever the filter you have provided, that you are going to see in the metadata line. Then further it is divided into header also. Then next, in that file you are going to get the data lines. So in the data lines you will get the entire information: what is the change of the base, on which chromosome it is present, whether it is a homozygous or heterozygous. So those all information you are going to get here in this VCF file. Okay, so that we are going to see in very detail like later on. But did you got an idea about this BCF and VCF file?
Yes,
Any doubt?
ma'am. Ma'am, if possible, could you please upload this note in our drive with
Yeah,
the
yeah, yeah, yeah.
definitions?
Yes, yes, sure.
Thank you. Thank you.
So next is the cigar string value, which tell you how bases from read is aligned to the reference genome. So for example, this is my reference genome, and on this reference genome, the read it is mapping, which is of 50 base pair here. So if 50 bases all are matching on this reference genome, that time my string value will be the 50M. So M stand for match here, okay, which will tell you all the 50 bases, okay, from this particular read it is matching to this reference genome. What if now only 40 bases are matching? That time you will get what? 40M, and there is a deletion of your base. So 10 will be your deletion here, and D is my, these are my operators. Okay, you can say associated operator, the M, D, I, X, that is the associated operators. Next, what if there is an insertion also? Okay, 40 bases are matching. It will denote 40M here. Then 5 is my insertion. So it is going to denote with the 5I, and 5 with the D. So D will, it is going to tell you that 5 bases are deleted here. Did you got this? Cigar string
Ma'am,
value?
could you please repeat this thing
Yeah,
again?
yeah, yeah. See, this is my reference genome, and cigar string value means how bases are aligning to your reference genome here. So in this figure, you can see this is one read which is of 50 base pair here. In the first example, what we saw, all the 50 bases here on the read, it is aligning on your reference genome, and it is giving you 50M. That means all the 50 bases are matching, okay, on your reference genome. Second is, if only 40 bases are matching, that means 40M here, and 10 bases are deleted. So it will denote with the 10D here. Next example, 40 bases are matching here, then it will going to tell you 40M, then 5 are insertion, okay, then 5 are deleted from this particular read. Did you got this now?
Yes, ma'am, understood.
Next is the fast queue format. So you are going to get your raw data in this format, FASTQ format. So FASTQ format is the 4-line format. That means 4 line is equal to 1 read. For example, there is 40,000 lines. So can you tell me how many reads will be there?
$10,000.
Moira, what about you?
Repeat the question, ma'am. I couldn't get the question.
4 line is equal to 1 read. If you are having 40,000 lines, then how many reads will be there?
10,000, ma'am.
Correct. 10,000 reads. Now, what those 4 lines, the information it is having. So first line, it is getting started with the at the rate, then sequence, it is giving you ID. Okay, it is giving you the ID here in the first line, and in the second you are going to get the actual sequence. Third line, it is a separator line. So for example, you have used the Illumina platform, it is going to take this as the plus sign. Then this plus sign, what it do, it just separating the second and the fourth line. So fourth line, it is having what? The ASCII characters, which represent the Fred quality score. So in this, you are going to get the information like this. Maybe okay. So this type of information you are going to get. These are ASCII character, which is just a Fred quality score. But can you tell me why the Fred quality score it is getting converted to ASCII character? So there is one method, that is the encoding method. So with the encoding method, it is going to convert the Fred quality score into the ASCII score. Why? Because as you can see here in the FASTQ file, 4 line, it is having what? It is equal to 1 read. That means it is a huge file here, right?
Yes, ma'am.
In order to reduce the file size, it is just converting this Fred quality score to ASCII character. Okay, with the encoding method. So did you got this all the terms in this NGS? Any doubt?
Yes, ma'am.
Okay. So now we are going to begin with the raw data first. So first step in the DNA sequencing is to download the raw data. So just open the NCBI SRA. So from this database, we are going to download the raw data. So for example, my application name is what? The DNA sake here, right? So what you have to write, you just write your application name. So for example, I'm writing DNA sake, and if you want to be more specific, you can just put any operator here and type variant calling, then search it. Okay, now you can see here there are some filter present. Assess on the left hand side, there is a controlled and the public. So control access is what? Whoever has submitted data, they don't want to share freely. So we have to ask the permission or for the person to download the data. Whereas in the public, you are getting only the free articles here. Okay, there is a freely available source here in the public. You don't need any permission for this. Now as you are putting the DNA sake here, you can see the source is only the DNA here, and total entry is this much, you can see. Next is the type is exome here. Then layout, we have already discussed whether it can be the paired end and the single end. And there are sequencing machine platform. Again, you can be very specific if you want to get a data from Illumina and Torrent. It's again up to you. Then strategy exome here. Then this data, it is also storing in the cloud also. And you are getting the file type as the BAM. In the BAM file, you are getting all the mapping information, and the FASTQ is your raw data. Okay. Now never download the data from the first page. Why? Because still the research is going on for this particular data, and you are not going to get a complete data for this. Okay, so always prefer the older data. So you can just open any one of this link. Now again, you can see here it is giving you, this is just a lab experiment ID here. You can see the title. Okay, then Illumina platform they have used. So one time they have used, you can see one time the Illumina has been run here. Then you can see how many spots in the Illumina they have used, and the total size of your data, you can see that is the MB here. And the design, you can see, then it is submitted by this university. Then if you want to read more about this article, you can read in this abstract option. Sample is your, it is has been taken from human. Then again, you want, this is your single end data. Okay, so what if I want to download this data? You have to go with this run ID. And you can see, just select the fast here, and you are going to download the FASTQ file here, right? This file. So this is about your single end data. But what if you want to download the paired end data? So that time you can just select this paired end data from here, and then again go to the older page. Then you can select any one of this. You can see this is your paired end data. And if I want to download this, I'm going to select this run ID from here, FASTQ. So you can see here this run execute. So here in the paired end data, what happened? We have already discussed in this, in the terms also. You are going to get two files, right? If you remember in the paint also I've discussed, you are going to get the two files here, id underscore 1 and id underscore 2, right? But in this NCBI SRA, this is one of the drawback. You cannot download the paired end data from this. Why? Because it is not going to give you separate two files, okay? Forward and the reverse file. So it is very difficult for us to separate the forward and the reverse file, right? For the paired end data. So what you have to do, you just simply copy this ID and go to the ENA browser, and you can put this ID and view. Same information you will get, and you can see underscore 1 is your forward and this underscore 2 is your reverse file. So from here you can download the paired end data. So did you got this? Any doubt?
Ma'am, could you please show me again how did you enter the data in the ENA website?
Okay, so just open the ENA, then put this ID, run ID.
Okay, ma'am. Okay.
Yeah. So we are not going to execute this particular data. I will share the link. Just wait. So everyone, please open this particular link. I'm going to share with you guys also. So for this data, we are going to perform the DNA-seq. Just open that link. Did you open this?
Yes, ma'am.
Yes, ma'am.
Okay, perfect. So now this is our data. Okay. So you can see this is the targeted sequencing panel. Okay. And submitted by this department, and they have used the Illumina. Basically, in this abstract, you can read they are discussing about the TP53 variant here. Okay. So this is the main target, that is the TP53. We have to see what is the variant that is causing here in the TP53. So this organism is your Homo sapien, and the source is the genomic, you can see. And this is your layout, is your paired and data. So along with me, you have to perform. Okay. Just scroll down and you can see this is your run ID. Just simply copy this run ID. Copied.
Yes, ma'am.
Yes,
Then
ma'am.
you can go to the ENA browser and paste here. Now again you can see this is a Illumina platform, paired and genomic, and if you scroll down you are going to get the two entries because this is a paired and data. So _1 _2, right? So what to do? Simply just copy, right click it, and there is a copy link address. Copied.
Yes, ma'am.
Now just open the notepad and just write here step 1, downloading the raw data, and paste that link.
Done, ma'am.
Now this is your forward file, right? Same thing you have to do for your reverse file also. Simply just right click it, copy link address, and paste that in the notepad. Done.
Yes, ma'am.
Now before this link you have to type wget -c, like this. Done.
Yes, ma'am.
So what you guys can do, just create one folder in your system and just write DNS folder. I will write your batch name maybe. Created DNS folder.
Yes, ma'am.
Now this is the Linux command, okay? wget -c. So why I'm putting this -c here? For example, uh, in between my system is getting stuck, or and it might restart, okay, your system, or might be it is shutting down. So what happened after putting this wget -c, it is not going to interrupt, okay? So sometime if you download your files directly from this, it is going to download only incomplete information, okay, if your system is getting restarted, right? So with the help of this wget -c, it is not going to interrupt any downloading part. And how to download this file, you just simply copy this wget -c, this first link from here, copied,
Yes, ma'am.
go to your folder, DNSSEC folder, open.
Yes, done, ma'am.
Now from here you have to open your WSL, backspace it, WSL, enter.
Ma'am, I don't have Linux today.
Yeah, don't worry, ma'am. Just ping me before the session tomorrow. I will
Yes,
guide you for that. Okay?
yes, yes.
Yes. And then after that, you can download this raw data and
Ma'am,
yes.
I'm sorry to interrupt, ma'am. I couldn't, when I pasted it, I couldn't go to the WSL. Instead, it goes to the website.
No, no, no. You just open it again from here.
Uh,
Backspace. Can you share your screen, Moira? I will guide you. Wait.
just give me a moment, ma'am. I'll share my screen.
Yeah. Prajakta ma'am, don't worry. Um, the workflow, or you can say the pipeline, will be executed tomorrow only. Okay? From
Okay,
today,
ma'am.
we are just seeing just how to download the raw data, ma'am.
Yes, ma'am.
Yeah. Yes. Yeah. From up, open the WSL, Moira. Backspace it. No, 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 no. Type WSL here.
Okay, ma'am.
Mm-hmm.
Okay, now I have to paste the link here.
Then you have to paste that link. Paste. Mm-hmm. Enter. Yeah. Now you can see the forward file is getting downloaded here. Right? Now minimize
So,
it again, Moira, from here. And again open the WSL and then paste that second link, the reverse link.
is it okay, ma'am?
Yes, Moira. Yeah. So don't interrupt in between. Let it download. Okay, Moira?
Okay, ma'am.
So
Okay,
this
ma'am.
is how you guys are downloading the raw data. Did you got this? Any doubt here?
No, ma'am.
Okay. So you have to maintain this script also. Okay. Along with me. And so from tomorrow, we are going to see what? Any idea? After downloading the raw data? Quality check by FastQC. That we are going to see tomorrow. Okay? That whether your raw data is good or bad. That we are going to see in the tomorrow session. And you can also paste this link in your script, SRA link. Should I share this?
Okay, ma'am.
Yes, ma'am.
Just paste that in your notepad also. And what you guys can do, just save this file in your DNS folder. Okay? Or you can write here the name DNA sec pipeline and save this. Okay then. Any doubt? Anything you want to ask?
No, ma'am.
Prajakta ma'am, Akansha ma'am?
Ma'am, can I get the yesterday's lecture, which with the
Uh,
record date?
ma'am, it is already there in the drive. Do
Okay.
you have that SS?
No, ma'am.
Uh,
Ma'am, share the drive link in the group, ma'am.
Drive link. Okay. Okay, no problem. I will share that. Okay.
Yes, ma'am. Yes, ma'am.
Yes, ma'am. Okay, then we will end here. Then we'll see tomorrow. Okay, the fast QC step.
Okay.
Prajakta ma'am, please before the session, please ping me. Okay,
Yes, ma'am.
for the WS. Yeah.
Yes, ma'am.
Okay then. Okay, bye bye, guys.
Bye.